in them to make protein. And it's important to note that not all genes are expressed all of the time. There are about 75% of cellular genes that are constantly expressed. We say constitutively expressed. That is, protein is made using their information all of the time. We call these housekeeping genes. It's very much like when we keep house. There's certain things that need to be done every day, such as we need to make dinner every day, and we devote a fair amount of energy to doing that. On the other hand, there are genes that are regulated so that we only express them, we only transcribe and translate them when the cell needs their products. Okay, that's like, for instance, we make dinner every day, but we only power wash the gutters of our homes maybe once or twice a year. Okay, and the idea is that by selecting what it is that the cell wants to express or not, that allows the cell to conserve energy. That is, we don't leave the power washing machinery going all the time. We only fire it up when we need it. There are many, many ways to regulate gene expression. We are only going to touch on a few of those. We're going to talk about transcriptional control. Some texts refer to this as pre-transcriptional control. That is, do we transcribe the gene or not? Okay, and then we're also going to talk about translational control. That is, we transcribe the gene, but we only translate that mRNA that we made when we need it. Okay, these two methods allow a cell to respond quickly or even more quickly. So quickly would be transcriptional control. More quickly would be translational control according to environmental conditions. So as you can see from this slide, there are many ways for controlling gene expression in cells. For prokaryotes, we're going to be talking about operons. This is a method that is pre-transcriptional regulation, although you'll hear me refer to it as control at the level of transcription. That's how I was taught, and I occasionally lapse back into that. We'll also be talking about um, riboswitches, which we see in prokaryotes, and some fungi. And then we'll be talking about targeted regulation of gene expression, that is using small interfering RNAs or RNAi, which is potentially quite interesting for its therapeutic implications. We'll talk about that more in just a bit. So operons are functional units of genes. These are found mostly in prokaryotes, and these were the first uh, elaborated, discussed ways of controlling, regulating gene expression. Um, the discovery of operons and figuring out how they work earned Jacob and Monod a Nobel Prize. Okay, so what we have with an operon is we have a series of genes that are clustered together on the chromosome. And so we will have structural genes. These are genes that code for protein. On this slide, the structural genes are Z, Y, and A. Okay, immediately prior to those, we'll have a control region. We'll have a promoter. Okay, and we know that the promoter is where RNA polymerase binds to set out transcribing a gene to making mRNA from it. Upstream from the control region, okay, that is to the left of the P, we will have a gene for a regulator. When we talk about operons, we need to distinguish between the two main types of operons. We have inducible operons. That is, the genes are all silent, they're turned off, they're not being transcribed unless they're activated by an inducer. Therefore, transcription must be induced. An example of an inducible operon is the lactose operon, frequently referred to as the LAC operon. Our second type of operon is the repressible operon. Okay, repressible operons, the structural genes are going to always be transcribed, so they're always on unless they're repressed, unless they're deactivated by a repressor. So we say then that transcription must be repressed. An example of a repressible operon would be the arginine operon, also referred to as the ARG operon. In general, inducible operons typically contain the genes whose proteins are going to be used in catabolism, okay, degrading. In contrast, repressible operons, the structural genes, are typically going to be ones where the proteins are needed for anabolism, for synthetic pathways, for making things that the cell presumably always needs. Let's look. So we're going to talk about the lac operon first. So we're going to talk about the lac operon first. The lac operon, the structural genes are the ones that the proteins are needed for catabolizing, digesting, degrading lactose. Okay, so the key to this is the regulatory gene, the I, which encodes a repressor. Okay, that's that blue, slightly crescent-shaped figure. Okay, now in an inducible operon, the repressor is being transcribed and translated, and can't go. Inducible operon, the repressor is being transcribed and translated, and the repressor is always made, and it's made in an active form. Okay, what that means is that the repressor, after being made, can go and bind to a downstream region called the operator. Okay, and if the operator has the repressor bound to it, then it's like a roadblock so that RNA polymerase can bind to the promoter, but it can't go anywhere because it's blocked by the repressor in the operator. Okay, it's rather like when one has a boot placed on one's car. Well, the car would work, but it's not going to go anywhere because the boot won't let the wheels turn. In this case, the repressor bound to the operator will not allow transcription to proceed. Okay, this is what happens when lactose is absent and glucose is present. The cell would much prefer to use glucose. Enzymatically, metabolically speaking, it's simpler for a cell to use glucose than it is to use a disaccharide like lactose. Okay, now, so the operon is off. If we have lactose present and there's no more glucose, well, then the cell says, well, uh-oh, this is what I've got to eat. It's time for me to switch on my lac operon genes that I need for utilizing these lactose so that I don't starve to death. All right, so what happens is that we've got the repressor. It's always being made. It's always made in an active form. That is a form that can bind to the operator. However, if we have lactose present and no glucose, then what happens is a certain amount of the lactose is turned into a form called allolactose. Remember, allo means other. Okay, so it's chemically different from lactose, and what it will do is bind to the repressor. That will prevent the repressor from binding to the operator. Okay, and so now when the RNA polymerase binds to the promoter, transcription can proceed through the now cleared operator region and into the region where the lactose utilization genes are located. Okay, and then we have the RNA for those, and now we can make protein by translating that. Okay, and I think we need to think of it in a way of, let's say that we're E. coli, and we know that E. coli is found in the guts of mammals. One of the definitions of being a mammal is that we're milk fed when we're young. Okay? And in many cultures, after children are weaned, they no longer consume milk. This is notably true in cultures that are non-herders. That is, they don't keep animals like cattle and sheep and goats to herd. Okay, so it makes no sense for them to be able to deal with lactose in their adulthood, so they can't, and their E. coli don't. Let's think about this a little more. Okay, so if you're an E. coli in the gut of a young mammal, then lactose is going to be present because you're milk fed, right? So one would then want to have the lactose operon on and be able to make the products that one needs for catabolizing the lactose. Okay, now picture one being an E. coli in the gut of an adult in a non-milk consuming, a non-dairy consuming society. Well, you're not going to get any lactose. So it makes sense to switch those genes off. Why make stuff that you don't need? Okay, unless, for instance, one happens upon some cheese or something like that and then eats it, and now the E. coli can go to work on it because they have their uh, lac operon being operated again. Okay, let's look at the ARG operon, the arginine operon. Okay, and this particular operon contains structural genes that have the information for making the enzymes that are necessary to make the amino acid arginine. Okay, amino acids we know are components of proteins. Cells constantly need proteins, so typically they're going to constantly need arginine. Okay, so in this case, the arginine operon is on, and the repressor is made, and that's that blue figure again. But instead of being made in an active form like it is with the lac operon, it's made in an inactive form. Okay, and so the genes for arginine manufacture are able to be transcribed by the RNA polymerase because there's no roadblock in the operator, and then the cell can go on to continually make arginine, which gets siphoned off into protein manufacture. Say because so the arginine never builds up to high enough levels to act as a repressor. Okay, now, what if the cell is entering a phase, perhaps it's going to become dormant and so doesn't need to make protein, and so it's not siphoning off the arginine that's being made using the ARG operon. So we get a buildup of arginine. That then activates that repressor, and now it unfolds and goes to bind into the operator, which acts as a roadblock. Okay, that is the arginine operon has been repressed. All right, so we've been looking at control at the level of transcription, that is, whether genes are transcribed or not, whether we make an RNA. Okay, at that level, it requires, in order for us to get the product of the gene, the protein, two steps. We have to transcribe the gene, and then we have to translate the mRNA that we made as a result of transcription. That is going to take a cell more time to respond to environmental change. What if a, what if a cell needs to respond more quickly than that? Why don't we cut out one of those steps? And this is what control at the level of translation will do for us. It's going to allow rapid response to environmental variation. An example of translational control would be that of riboswitches. Okay, and so what we have is we have a gene that's at the top of the page, and we're going to transcribe that. Now, this gene is one that has a riboswitch sequence. Okay, and the riboswitch sequence in the dark orange is non-coding. Okay, and it's before the protein coding sequence. And so what will happen, because remember, RNAs also fold, they have three-dimensional structure, is that the non-coding sequence will fold back onto the protein coding sequence and block it so that the translation machinery, the ribosomes, can't access the protein coding sequence. Okay, so we've made the RNA, but it's in an inactive state unless certain conditions are met. In this slide, what we're seeing in the figure is that there is a target chemical that the genes of the protein coding sequence, that is actually the protein product of the protein coding sequence, are needed to deal with. So that target chemical will interact with the riboswitch, causing it to unfold to reveal the peach colored, the light orange area, which is the protein coding sequence of the mRNA. Okay, when that sequence is able to pop out away from the non-coding sequence, what that's going to accomplish is that now the translational machinery, the ribosomes, will be able to access the protein coding sequence, and now we can make the proteins that we need to deal with the target chemical. Other conditions that can activate riboswitches could be pH, could be temperature, or various other things. Now, and it's an interesting aside, um, we find riboswitches strictly in bacteria primarily. I shouldn't say strictly, but mostly in bacteria. They are in some fungi and plants, but we don't seem to find them in animals. Okay, and we've talked previously that when we're trying to devise antibiotics, chemotherapeutics in our fight against pathogenic bacteria, what we need to do is target things that the bacteria have that we don't. Okay, target differences between the prokaryotic cells and the eukaryotic cells. And we are rapidly running out of antibiotics. I'm sure you're aware that antibiotic resistance is a growing problem clinically. And we really have only a very few targets for antibiotics. For instance, targeting the prokaryotic cell wall or targeting the prokaryotic 70S ribosomes. So we desperately need new targets. Potentially, riboswitches could be targeted. And I've got the dollar signs because whenever one develops a new drug, particularly one that fills a uh, niche where there's a certain amount of desperation, okay, that is a resistant microbe, then one can charge what the market will bear. All right, so another method of controlling gene expression at the level of translation, that is translational control, is that of short interference RNA or siRNA. Learning about and understanding the method for this occurring won its discoverers a 1998 Nobel Prize. So what we have going on is in step one, we have for instance, virus gene being transcribed in a host cell. So we have this RNA transcript, which is processed into a mature mRNA. And now the cell can produce a short interference RNA that's complementary to part of that abnormal mRNA. In step two, that short interference RNA will bind to the abnormal mRNA. Now, cells don't like double-stranded RNA. To cells, what that means is that there's a virus present. And so cells have a mechanism called an RNA-induced silencing complex, the RISC, that will take up that double-stranded RNA and then chop it up, just break it down. And so now that is no longer able to be translated. And so the gene or the genes involved are what are called silenced. Okay, now the short interference RNA can be made either by the cell or it can be artificially introduced. This a few years ago was a hot area of research because what it potentially could do is allow targeting of any gene, specifically the RNA produced from that gene, and turn it off selectively. This was thought to have promise in things like treating high cholesterol disorders. It has been used experimentally for treating Ebola virus infection in primates. One of the biggest problems with this method is actually introducing the short interference RNA, the artificial short interference RNA, into the cells to be treated.